Hi, welcome to Chapter 9. We're learning all about genes and inheritance. And this is Dr. Randy Pepke, your guide to the information, at least for the next hour or so. So, genes and inheritance. Let's start out with a basic definition of heredity. Heredity is the passing of characteristics from parent to offspring. And this happens through the parent's genes. Now, we do know that some traits are controlled by a single gene, but when we look at people who, father and son, we we see a lot of traits that are not controlled by a single gene, and yet the father and son, they look very, very similar to each other. So Jaden Smith looks very similar to Will Smith because of genetics, but a complicated set of genetics that we unfortunately don't get to hit too much of. We're going to stick to the simplest types that we can. Even talking about the simplest types, though, it's still fairly complicated. And, human beings have been noticing that fathers and sons look a lot alike ever since we've been able to notice anything, really. But it wasn't until we started selectively breeding animals and plants that we started to see how easy it was to observe heredity. Thousands of years were spent looking at animal and plant breeders, understanding there was a connection from parent to offspring, but really not getting what it was, how it worked. And even back in ancient Greece, they bred horses, maybe not these tiny little horses, but they definitely bred horses. Once breeders started to see heredity exists, they started doing the selective breeding. And again, size in horses, sure, but sweetness of corn, or loyalty in dogs, or how docile a cow is, or how beautiful a flower is. So it turned out that a lot of these practices were very successful and so forth, but we didn't really understand how it happened. So these days, we do have a lot better understanding of it, of the way heredity works. And as I hinted at before when I was talking about Will and Jaden Smith, that single genes are one of the ways that we know that heredity is passed on. Remember, a gene is something that codes for a protein. And there's different versions of those called alleles, right? We've already kind of covered that in this book, in this semester. So again, if you focus on a single gene, it actually becomes easier to understand how these breeders did what they did and how, in fact, heredity works in the first place. So, so some things that are single genes are the length of fur in cats. So a long hair cat versus a short hair cat. We just have dominance and recessiveness here. And in fact, we also have coat color in cats. And at least for some of the simpler colors, like white versus not white, again, it could be any color, but white versus not white, we have a simple example of dominance and recessiveness. When it comes to humans, there are a lot of genes, and a lot of them are complicated, like I talked to you about before, but there are some that are really simple. For example, Huntington's disease is a well-known disease where if you're dominant for the trait, then you will have the disease, and if you're recessive, you won't. So it's just a single gene with a dominance and recessiveness. And there's a few others along those lines, but again, the majority of inheritance in humans and other species as well are just really complicated to understand. So what did people used to think before we had our modern understanding of genetics? And our modern understanding of genetics was first discovered by this dude, Mendel. He was a monk. We'll learn about him in a second. But there were lots of misunderstandings about how heredity worked. One of them that is just kind of fascinating to me, in your book as well, is that there was this mistaken idea that inside every little sperm was an actual baby, a tiny pre-made human baby, and that baby was implanted into the woman's uterus, just by this act of sperm getting inside the woman's body through sex. So you can kind of understand some of the misconceptions that are raised by this, like any genetics that would exist would only exist from the father. And what does the mother contribute to an offspring? You can also see in this a lot of, well, not a lot of diversity in thought processes. This is a thought process from a very male-dominated society who, for a large part, scientists were from the upper classes of that male-dominated society, and so this is something that they kind of thought would be useful for passing on land, passing on money to the next generation. So this idea thrived for several hundred years before it was replaced with what the careful observations of Mendel. And there's Mendel right there, Gregor Mendel, the monk that I told you about before. His research was very critical for our understanding of heredity now. And so, sometimes when you do science, it's a lucky observation or something, that you noticed by chance. But with Mendel, it was actually very, very careful observation through methodical experimentation and scientific thinking. Scientific method totally came into play here. If it hadn't been for this kind of thinking, we still probably would not fully understand, at least in the way that we do now, with the words that we do now, the way that heredity exists. So he did his research by taking kind of three features into account. He picked a good organism, and he picked the pea plant, which they're very easy to grow and easy to maintain it and easy to breed. They reproduce quickly. You can get lots of generations in a single summer. So pea plants, he picked a good organism. I think he figured that he could apply what he learned from plants to any kind of animals. So that's the first thing. The second thing is that pea plants have very easily categorized traits. So unlike humans or unlike dogs or cats or even mice, it's really easy to say for the color of the flower, it's either white or purple. There's really no in between there, white or purple. The shape of the seed in the pea pod, the shape of the seed, is either smooth and round or like shriveled up, wrinkled. There's really nothing in between. It's either one or the other. The color of the seed, of the actual pea, can be either green or yellow. And again, nothing really in between. There's some other ones too that he looked at, like he looked at the color of the pod and the pod shape and the location of the flower on the plant and how tall the plant was. But those are some really easy ones for you guys to understand. So the last thing that he did was that he was able to breed them into several distinct populations. So he had populations that were only white flowered. He had populations that were only purple flowered. He did this for each of the categories that he was looking at. I don't know if you can imagine the size of the garden that must have existed for Mendel to have established all of these different, just so many numerous distinct populations that were kept separate from each other. And when he was doing this, he called them true breeders, because every time he bred a white one to a white one, he got only white offspring. He called these parents true breeders. And the same thing for the purple. If he bred two purples together and he only got purple offspring, he considered these ones true breeders, meaning they bred true to color or true to form in the case of these other ones. So he had this really strange result that happened when he actually started to do his crossbreeding, when he bred a purple to a white. And when he first did that, in the first generation, what he got was 100% of them were purple. And so that was a bit strange. because he didn't expect that, you know, two true breeders of opposite phenotype. And remember, we learned the word phenotype before. It just means outward appearance. In this case, we're talking about the color of the flower. And so he took two for those very distinct populations. Remember here, he took one from here and he took one from here and he crossbred them. And he got all purple. And this is where he started like scratching his head thinking about this concept of dominance and recessiveness, which I've used this word with you just a few minutes ago in this lecture. So what Mendel realized is that some traits will mask other traits. And he used the word dominant to describe a trait that did the masking, in this case purple. And he did use the term recessive to describe the trait that was masked by the other trait. So dominant versus recessive. Because in here, I mean, it's clear that they must have gotten, these offspring here must have gotten one kind of set of information from the white one and one set of information from the purple, but we're not seeing the white information. But then he did something really interesting and important too. He took these two of these offspring here, which he named the F1 generation. This is the parental generation up here at the top, true breeders. There's parental, not the same thing as parents, because when he was in this F1 generation, he took two of the F1s and made them parents. Again, not parental. Parental is two true breeders of opposite phenotype. So here we have these two non-true breeder F1s that we know they're purple, but we don't know anything about them. And he crossbred those. And what he got was just like kind of mind-blowing to him. About 3/4 of the offspring showed the dominant, but there pops up the recessive ones, the ones that had been masked in this generation. They show back up again. So how was it possible to get the white plants if he was breeding two purples? And again, once again, he realized that the white was recessive, and that these two purple over here, each of them have a dominant allele that's showing, but they also have a recessive allele that's not showing. And so when you cross those two together, some of those will get two recessive alleles, and they will show because they don't have a dominant allele masking them. So the three important things to get out of Mendel's research is that each parent, you don't put all of your information into your sperm or eggs. You put half of it, one single set of genes into each sperm or egg. Now we learned that when we were talking about meiosis, but Mendel understood this not from understanding cell division. He did it from looking at crossbreeding right here. Same conclusion, different methods. The second thing that we get out of this is that the offspring, the offspring from the original cross, they're going to have two copies, one from the male parent and one from the female parent. Now, I know you don't think about flowers as having male or female, but we do have pollen and we do have eggs, and you can think about the parts of the flower as male or female, if that helps. Finally, we have the trait that you actually observe is dependent upon which two copies of the gene that you got from your parents. We know that you could have two of the same copies from the parents, from mom and dad, and in that case you would be homozygous. If you had two different versions of the alleles, then you would be heterozygous. Those are terms that we learned about in lab a couple weeks ago. If you have two dominant alleles, then you're homozygous dominant. And if you have two recessive alleles, then you're homozygous recessive. Heterozygous is going to have one of each, so you don't say heterozygous dominant, even though they might be showing the dominant phenotype. You just say heterozygous, with the assumption that, of course, they're going to show the dominant allele, and the dominant allele is going to mask the recessive allele. So this is putting into like a visual thing for you, the alleles and how those alleles get passed down to each generation. This particular one is a cross between two heterozygotes. Notice the purple versus the white here, and the purple versus white, two different alleles. That makes them heterozygous. We could also say that maybe we were talking about this generation. That's the F1 generation. So when you go through meiosis, you segregate or separate homologous chromosomes. You do that during meiosis 1, remember? But Mendel didn't know meiosis 1 or meiosis 2. We're just kind of integrating everything together. And of course, this parent over here is also going through its own segregation of the purple allele from the white allele. And those alleles are on those homologous chromosomes, but they're segregated. They're put into different eggs or different sperm. So if we're talking about fertilization, the next thing is, this kind of gets really messy here, right? This is really actually hard to follow. You could have this white allele from this parent joining up with this white allele from this parent, and you'd say, oh, you can make a homozygous recessive offspring, and like follow the lines. Or this purple allele could join up with this white allele, and you get heterozygous. Or this purple could join up with this white, and you get heterozygous. So there's actually two different ways to get heterozygous. Yeah? Oops, sorry about that. Two different ways to get heterozygous. There's only one way to get the homozygous recessive. You have to get one from mom and one from dad. And the same thing is true for homozygous dominant. You have to get one from mom and one from dad. But for the heterozygous, again, two different ways that you can get that heterozygous. So Mendel called this idea where the parent separates the alleles into different cells. He called that the law of segregation. And so that's what we are going to call it, the law of segregation. So it turns out that, things aren't always as they appear, right? If you take skin color in humans or any mammals, you can see that, that melanin is what produces skin color. And of course, we have different gradations of skin color from very dark to very light. But the fact that some individuals can make melanin, like you see with this deer, and other ones that really can't make melanin, like in this albino deer, is something that is based on genetics, and that is very clearly influencing the outward phenotype. Now, again, if you're looking at human skin coloration, you wouldn't just be able to look at somebody and say, oh, you have this version or this version or this version. But what we do know is that for anybody that can make melanin, anybody except an albino, that they are in fact have the ability to make melanin. And so their phenotype in this case would be melanin producing. This deer can make melanin. Unless you're albino, you can make melanin. This albino deer cannot make melanin. And those are the phenotypes, the outward appearance. Notice in the deer that even the eyes don't have any melanin in it. They're very light color. The ears don't have any melanin in it, not just the fur color. So again, phenotype can be the outward appearance, just looking at something. But phenotype is not only about what you can see. It can also be less easily visible characteristics, like the chemicals that you produce for whether you can make blood clot or not. We talked about that in earlier labs, right? You could think about whether or not you can digest lactose without being uncomfortable in your guts. So those are also phenotypes. You could think about phenotype as a behavior. All of those things are observable in one way or another. The thing that underlines a phenotype is the genotype, and that's the genetic composition. When we talk about genotype, we always reference a specific trait, and we always reference the type of alleles that they have, always. So in this case, we do know that albinism is recessive. So this deer right here, we could very easily say has the genotype homozygous recessive. Little a, for example. Phenotype albino, genotype homozygous recessive. We look at this deer right here that makes melanin. Do you know the genotype of that one? Not really. It could be heterozygous or could be homozygous dominant. You just don't know. But in general, the word genotype refers to the specific allele combinations for each of the phenotypes that you might see. So let's take a look at a couple of crosses that your book does. And this is in direct opposition to the very confusing version of it we saw here, where we're trying to follow the arrows and it became a little confusing. The Punnett square is actually a solution to that confusing way of drawing arrows. So what you do with a Punnett square, and we will spend a lot of time in lecture dealing with Punnett square and lab as well. And I'll have some very specific rules that I'll want you to follow. I'm not going to talk about them in this lecture, but I will talk to you about them in lecture and lab time that we spend together. So, and I think there might be a separate video that I can make on the rules that I'm expecting you to learn. But again, this is just from your book, and I kind of want you to learn this to start out with. Okay, so if you have a true breeder, a homozygous for melanin, and this father giraffe right here, and you have this mother giraffe that is homozygous for albino. Of course she's homozygous for albino. Remember that if you're recessive, you have to be a true breed. You have to have both of your alleles be recessive. There's no other way. Otherwise, you'd be showing melanin. So we got, your book uses the letter M. I think the letter M is a terrible letter to use because it's hard to tell the difference between big M's and little m's when you're writing your notes. But we're going to go with what the book is using. And so little m for homozygous recessive and big M for homozygous dominant. Remember that we go through segregation, so we would separate those two M's into different cells. I like how your book draws little circles around them, like they're little gametes. And you put the gametes, little eggs or little sperm, in this case of the father sperm, onto one side of the Punnett square, and you put the little eggs from the mother on the other side of the Punnett square, and then you say to yourself, and you fill in the Punnett square, what would happen if the big M fertilized the little m? You'd get this heterozygote right here, big M, little m. And in fact, if you did that for all of the boxes in this Punnett square, you would get 100% of them being heterozygous. That's the genotype. 100% or all of them are heterozygous. The phenotype is all pigmented, all having melanin. So 100% is the way I would say that. 100% of the phenotype is melanin. If we take a look at another Punnett square where you take maybe two of those offspring and you mate them together, you have a father that's heterozygous and a mother that's heterozygous. And if you fill in the chart, following the law of segregation, separating the gametes, same here, separating the gametes from where you see from the mother, her heterozygous, one big M, one little m over there in separate cells. And then you say, what happens if the sperm and egg meet up? You get a giraffe baby that's homozygous dominant. But if this dominant M meets up with this recessive M, you still get a pigmented giraffe. It still looks the same. The phenotype is still the same. But notice the genotype is different. Homozygous dominant versus heterozygous. Also notice that there's two different ways that you can get heterozygous individuals. You can get the father giving the little m and the mother giving the big M. Or you can get it as the father giving the big M and the mother giving the little m. If both little m's are given to the offspring, then you have your albino. The genotypic ratio, and your book does this in fractions, and you can do it in fractions too, or you can do it as like a straight up ratio, but you get 1/4 homozygous dominant to 2/4 or 1/2 heterozygous to 1/4 homozygous recessive. That's the genotypic ratio. If you look at the phenotypic ratio, you have to combine anything that has the same phenotype. All three of these have the same phenotype. So 3 out of 4 are pigmented, and that's in ratio to 1/4 of them that are albino. So one of the things that I didn't talk about when we were looking at this is the idea that that you use capital letters to indicate the dominant allele, and you use lowercase letters to represent the recessive allele, and that is a kind of, I don't know what the right word is here, but it's just something you do. You always use capital letters of the same type of letters, so capital M versus lowercase m. It's convention, right? So what you would not do, what's not acceptable to do, is to say capital M versus lowercase a for albino. That would not be acceptable. You have to use the same kind of letter family. Sometimes you don't know what the genotype is. For example, if I go back here to these purple flowers up here, you don't really know if this one's homozygous or heterozygous. And if you're trying to solve a puzzle for a Punnett square and you're not directly given the genotype like we have been here, sometimes it's nice to use like a little dash as a placeholder. Like maybe you didn't know that this one was heterozygous, so you'd say capital M. I know it's capital M because it's got melanin, but I'm not sure if this is lowercase m or uppercase m. You can put like a little blank as a placeholder, and that placeholder is like an unknown, which you can fill in later after you solve the puzzle. So that's the basics of how to do a Punnett square. Again, we'll get lots of practice of this in class. Tay-Sachs is a deadly disease that's associated with lysosomes not working properly. And it's a recessive disease. So what I'm going to teach you here is how you might solve a problem of dealing with how to figure out what the chances of inheriting a malfunctioning gene where you could potentially end up having an offspring that has the deadly disease. And again, Tay-Sachs is deadly. You're talking about death in childhood, early childhood, around age 2 is common for when children with Tay-Sachs die. So if both parents are heterozygous, and if they're known to be heterozygous, then each parent has a 50% chance of passing on that recessive allele. They have a 50% chance of passing on the dominant allele too, but we're really focusing on the recessive allele because that's what can end up with a child with Tay-Sachs. So 50% is the same as saying 0.5, right? Move the decimal point 2 places backwards and you get 0.5. Same thing for the other parent. The other parent who's heterozygous also has a 50% chance of passing on that recessive allele to that offspring. And so 0.5 times 0.5 is equal to 0.25, which means that the child has a 25% chance of having the recessive disease, the recessive disorder, Tay-Sachs. So two parents who are heterozygous have a 25% probability that they will have a child with Tay-Sachs. And so if a couple only has one child and they're not sure if they're homozygous or heterozygous, you really couldn't use this type of probability to figure out what their genotype was. So you couldn't figure out that. But it is useful if you actually do know the probability for each parent. The next concept that we're going to talk about is something called the test cross, because I just suggested to you over the course of the last 2 slides that you don't always know what the genotype of a parent who is homozygous, or sorry, just if they show the dominant phenotype. You don't always know. They could be homozygous or they could be heterozygous. And so if you are trying to breed animals, in this case, I think your book loves the albino, so you had albino giraffes, now we've got albino alligators. If you, though, were living down in Florida and you had this alligator farm and you wanted to draw in a bunch of customers and you thought albino alligators would be a good moneymaker, then maybe you have this breeding program for albino alligators. But of course, you're probably not starting out with a whole bunch of albino alligators. You're probably starting out with some ones with melanin in them. And yes, it's still melanin. It's the exact same molecule as you find in you, in your skin color. So what you can do is something called a test cross. If you have one albino, in this case here, they've got the females, the albino, and we know that albinism is recessive. It's recessive in almost every organism. And you have the father who has melanin, but you're not sure if he's homozygous, big M, or if he's heterozygous, big M, little m. And so this is just a very interesting way to present a Punnett square. It's like two Punnett squares together. So if your male is big M, then all of the offspring would come out to be 100% having melanin, crossed with this with this albino mother. Now, on the other hand, if you're heterozygous, a big M and a little m, look what happens to the ratio. You still get half of them having melanin, but the other half will have albinism. And so this is why it's called a test cross. The only way you're figuring out what the genotype of this melanic male is by crossing him with a female that has albinism or is recessive. It's the definition of a test cross. Literally, you don't know the genotype of an individual showing a dominant phenotype, and then you cross that individual with the dominant phenotype to an individual with a recessive phenotype who we know must be homozygous recessive, because that's the only way you can show recessive. That is the definition of test cross. So depending on what the outcome is, you know that this father is either big M, or if it's 50-50, you get big M, little m. In humans, we'll talk about this on the next slide because it's about a half an hour in. So we'll stop the lecture now and restart up in just a moment. See you in the next video.